Hi everyone. In today's video, I quickly just wanted to go through the different types of neuroglia in the peripheral nervous system. Now, there are two main types of neuroglia in the peripheral nervous system. These are Schwann cells and satellite cells. Let's first start off by exploring Schwann cells. Schwann cells are cells in the peripheral nervous system that are analogous in their function to oligodendrocytes in the central nervous system. This is because they allow for the process of myelination. Now, myelination is a very important process in both the central and peripheral nervous systems, as it essentially allows for protection of the axon and also allows for what we call saltatory conduction. Saltatory conduction is a very important concept in the propagation of a nerve impulse. This is because essentially the formation of nodes of Ranvier that arise from this myelination allow for nerve impulses to jump across an axon. Say, for example, we have the cross-section of an axon, which is denoted by this circle here. A Schwann cell is able to wrap around this axon in a repeated manner similar to a Swiss roll. This wrapping around nature essentially means that the Schwann cell forms a multi-layered protective layer that is 90% lipid and 10% protein. So if we denote the axon in black, the Schwann cell will be the repeated structure that surrounds it. This structure contains a nucleus, which may be located somewhere here, and therefore allows for nerve impulses to remain within the next axon, and if a longitudinal section is taken with many Schwann cells covering a particular part of the nerve cell body, you will see that these sections, which I called before nodes of Ranvier, allow for nerve impulses to essentially jump across, and this allows for the faster propagation of a particular nerve impulse. Faster propagation basically means that the nerve impulse is able to pass in a faster manner over the nerve axon. The second type of neuroglial cell that is present in the peripheral nervous system is satellite cell. Satellite cells are not present on the axon, but rather are present on the actual nerve cell body or soma. So if we have the nerve cell body here with the nucleus in black, the satellite cells will be surrounding this nerve cell body, covering it as so. This covering has many different functions. Firstly, it allows for the protection of the cell. Secondly, they allow for the repair of the neuron if it gets damaged. And lastly, and one of the most important aspects of this neuroglial cell, in which it is analogous in its function to astrocytes in the central nervous system, satellite cells allow for the diffusion of nutrients into the neuronal cell body. So similar to astrocytes in the central nervous system in their ability to regulate which nutrients are able to pass into the cell, similarly, astrocytes are able to do the same thing. Therefore, in review, Schwann cells are analogous in their structure to oligodendrocytes in that they allow for the process of myelination. This allows for the protection of the axon as it is covered in a layer that is 90% lipid and 10% protein. This means that on a staining, it appears clear unless it is stained by lipophilic stains such as Sudan black. And this myelination allows for saltatory conduction as well, which is through the formation of nodes of Ranvier. These nodes of Ranvier are the spaces that are formed between the Schwann cells, which are denoted in this black, and allow for a nerve impulse to jump between these Schwann cells. The way in which this myelination process occurs is that the Schwann cells are able to essentially wrap around in a circular manner around the axonal body, and therefore allow for a multi-layered protection which contains a nucleus and also allows for repeated protection of the cell. What I forgot to mention is that in the case of unmyelinated axons, there is actually a Schwann cell, a single Schwann cell, that is able to myelinate many axons at once. So, if we have a Schwann cell that is structured like this, essentially, instead of it covering an axon body at once, it'll cover many axonal bodies. Essentially, it is able to wrap around, in this case, 5 axons at once, and this single Schwann cell is able to allow for their protection. However, these Schwann cells are only referred to as unmyelinated. On the other hand, satellite cells are cells that are neuroglial cells that cover the soma of a particular neuron. They cover it in order to allow for its protection, repair, and also the diffusion of nutrients into the soma, which is analogous in function to astrocytes in the central nervous system. I hope you've enjoyed this video. Thanks for watching.